Hello everyone, I am Janak Awasthi from Nepal. Warm welcome to you all on Janak Lecture Series. For more updates, please subscribe my YouTube channel at www.youtube.com/janaklecture. Hit a like on my Facebook page at www.facebook.com/janaklecture. Today in this lecture, we will be dealing with the introductory part regarding the human genetics. We'll define what is human genetics, we'll talk about the different branches of the human genetics, and we'll talk about the brief history of the human genetics, and we'll be dealing with the common terminologies regarding the human genetics, regarding the study of the human genetics, the terms which frequently do come across. We'll try to define those terminologies, but the specific terminologies regarding the individual chapters will be dealt separately. And we'll be talking about the principle of human genetics, and we'll try to classify the genetic disorder at the end. So what is human genetics? It is a science that deals with the variability and heredity in human being, and in the modern genetics it also approaches towards the genetic basis of the disease. So human genetics deals with these three different points. It deals with the variability and heredity in human beings, and newly it also deals with the genetic basis of the disease. So it includes these different areas like the medical genetics, clinical genetics, dysmorphology, cytogenetics, cancer genetics, and population genetics. Now we'll talk about these different branches with the help of the definitions. So medical genetics, it is a branch of a medicine which deals with the genetic disorders of medical importance. Clinical genetics, it deals with the diagnosis and care of patients with genetic disorders. Dysmorphology, it deals with the anomalies of morphological development which are associated with genes and environment. Cytogenetics, it deals with the number, structure, and function of chromosome by the use of a method that is known as karyotyping. Cancer genetics, actually the cancer is the multifactorial disorder. So the cancer genetics deals with the certain genetic components which are susceptible to develop a malignancy. But gene, it is not purely involved in cancer genetics. In some of the cancer, along with the gene, the environmental factors also do play the role. That's why it is a multifactorial genetic disorder. So population genetics, it is the study of the distribution of frequency of gene population and genotypes, whether they are maintained or changed. Now we are going to talk about the history of genetics. The history of genetics starts with the Darwinism theory of the origin of species in 1859. According to this theory, hereditary nature of variability is an important factor in evolution. So this was proposed by Darwin. And in 1902, Archibald Garrod, he discovered that in metabolism there are certain genetic factors involved. In 1902, William Johansen, he discovered the term gene. And in 1953, Watson and Crick, they discovered the molecular structure of deoxyribose nucleic acid. In 1956, total number of chromosome was discovered, and it was mentioned that the total number of a chromosome in normal human being, it is 46. Out of them, 22 pairs are autosome and 1 pair is sex chromosome. But before this, all of the studies regarding the counting of the number of chromosome, they were based on the samples derived from the patient. That's why the number was argued to be variable, whether it is deviating from 46, it may be 45 or 47, and there was an argument ongoing. But later on in 1956, extensive study was done in the normal population, and it was mentioned that the total number of a chromosome in human being, it is 46. And in 1968, Indo-American scientist Hargobind Khorana, he discovered the genetic code. In 1970, the molecular genetics, it is the branch that kept on evolving, and in 1990s the human genome project started, and it was completed during the early 2000s. Now let's talk about the common terminologies used in the human genetics. The first one is the human genome. The human genome, it is the complete set of genetic information encoded as DNA within the 23 chromosomal pairs present in the nuclei and mitochondria. So we can see over here, this is the helical structure of a DNA which winds around the histone protein and forms a nucleosome, and this nucleosome undergo folding to form a chromatin fiber, and this chromatin fiber undergoes extensive folding, we can see over here, and it forms a solenoid, and this solenoid again undergo coiling to form a loop of a chromatin fiber, which are condensed to form the chromosome. So this is how the genetic material, it is present within the nuclei of the human cell, and the genetic basis or genetic material is the deoxyribonucleic acid, which undergo coils and series of folding to form a chromosome. And this chromosome, it is very much condensed during the metaphase stage of the cell division and can be visualized under the light microscope with the help of the proper staining. And there is another terminology, that is karyotype. The karyotype, it is the number and the visual appearance of the chromosome in the cell nuclei of an organism or a species. And there is a similar terminology, that is the karyogram. It is also called ideogram, and we can see this is the ideogram of the human being. It is a diagram or photograph of the chromosome of a cell arranged in homologous pair and in numbered sequence. We can see the chromosomes are arranged according to number, and this is the arrangement that is done on the basis of the decreasing height of the chromosome. We can see the homologous chromosome in pair, the chromosome number 1, 2, 3. So in this way, the autosomes up to 22 number, they are arranged in the decreasing height, and the sex chromosome, they are placed at the end. So this is the karyogram of a male having 22 pairs of autosome and 1 pair of sex chromosome, and this sex chromosome is placed at the end of the picture. And this pictorial view of the arrangement of a chromosome present in the cell, it is known as karyogram or ideogram. Now let's define gene. The gene, it is the basic physical and functional unit of heredity made up of DNA, which act as instruction to make protein or RNA molecule. It is not always mandatory that the gene, it is encoded as a mRNA, and mRNA encodes the rRNA, and that encodes the protein. So that is the common central dogma, where the gene is finally encoding the protein, but sometimes the gene may encode the RNA molecule, which act as a regulatory molecule in the chromosome. And in human being, gene may vary in size from few hundred DNA bases to more than 2 million bases. And in human being, after the Human Genome Project, it has been made sure that there is a presence of about 25,000 genes. Now, talking about the other terminology, let's define what is locus. It is a specific position or location of a gene on a chromosome. And allele, it is the alternative form of a gene. And this allele, it doesn't mean that it is a mutant form of a gene. It is just the alternative form of a gene that is present on the homologous chromosome. An exon, it is the transcribed region of a gene that is present in mature messenger RNA. An intron, it is a segment of a gene that is transcribed initially but removed from RNA transcript. So when we see over here, this is the segment of a gene. With this segment of a gene has got the promoter region, we can see over here, and the gene includes the expressing segment, these are the exon, and the inexpressing segment, that is the intron. So we can remember it with the help of a mnemonics. EX is, we can use it as an expressing segment, and EN we can use it as a inexpressing segment. So EIN for inexpressive, EX for expressive. So exons are the expressive segments, and introns are the inexpressive segments. And later on, during the formation of the mRNA, after the replication of after the translation of this gene, these introns are removed and exons, they ligate to one another and form a transcript, RNA transcript or mRNA. Genotype, it is the genetic constitution of an individual. Phenotype, the observable characteristics of an individual is phenotype, which is determined by the underlying genotype. Dominant, it is the expression of one of an allele, and it is always present in or expressed in heterozygote. Recessive, it is always expressed in homozygous condition where for its expression two alleles are necessary. Mutation, it is the any permanent heritable change in the sequence of genomic DNA. Epigenetics, it is defined as the factor that can affect the phenotype without change in genotype. So epigenetics includes the environmental factor which can affect the phenotypes. Now let's talk about the principle of human genetics. The human genetics, it undergo in progression after the discovery of the Austrian monk Gregor Mendel in 1865, but his work was not accepted by the scientists throughout the world, but later on after his death, by the independent experiment by all the other different scientists, they concluded that the work of Gregor Mendel was true and it was accepted as the principle of the human genetics. So Gregor Mendel defined the three different principles of the human genetics, which includes the law of dominance or unit inheritance, law of segregation, law of independent assortment. So law of dominance, it can be defined as, according to law of dominance, each trait or character is determined by two alleles inherited from each of the parents. These alleles may exhibit as dominant, codominant, or recessive expression. Dominance, it is present in heterozygous condition, and in codominant condition both of the alleles are expressed. Like the best example is the blood group of the human being. And in recessive condition, both of the allele in the different location of the homologous chromosome must be expressed for the phenotypic expression. Law of segregation, it is the second law by the Gregor Mendel, according to which each of the two inherited alleles will segregate and pass into separate gamete during meiosis, and each gamete carries only one of the allele for each character. But slight exception to this law of segregation is that the adjacently lying gene, they are not segregated, they pass togetherly. And the best example is the hairy pinna, and this hairy pinna that is present throughout the generation. It is because the genes of causing the hairy pinna, they are adjacently lying and they are not segregated during meiosis, and they happen to pass from one generation to another generation. That's why we can see the hairy pinna in every generation, and that is slight alteration of the law of segregation or exception to the law of segregation. And next is the law of independent assortment. According to this, in the gametes, allele of one of the genes separate independently of those of the another gene. Thus, all possible combinations of the allele are equally probable. Now, let's classify the genetic disorder. The genetic disorder can be classified into the classical form of genetic disorder and the non-classical form of the genetic disorder. The classical form of the genetic disorders are single gene disorder, which is also called as Mendelian inheritance or Mendelian disorders. And next is the chromosomal disorder, which are associated with the changes in the structure or a number of a chromosome. And the third one is the multifactorial disorder, in which both the genes and the environmental factors are involved to cause the disease. Whereas in the single gene disorder, there is the alteration only in the single gene, and that causes the genetic disorder. And the next one is the non-classical form of the genetic disorder, which is because of the alteration in the mitochondrial DNA. So it is the mitochondrial disorder. And this mitochondrial disorder, they are always maternal in origin, because during fertilization, the mitochondria which are present in the neck region of the sperm, they are all degenerated. That's why all the mitochondria, they are derived only from the ovum. That's why all the maternal mitochondrial disorders, they are derived from the mother. They are maternal in origin. For example, Leber's optic neuropathy. So with this, we come to the end of our lecture. Thank you very much for watching.